right, good afternoon. Welcome back. We have 6 lectures to go. And then you're finished with me forever. Well, not forever, no. You have my email. You can always reach me, but the course will be over.
Follow you on YouTube.
You could do that. Yeah, I'll be there. All right, so today's the second half of dealing with an infection. We're going to talk about antivirals. And these, what is an antiviral? It's a small molecule. What is a small molecule? It's not a protein. Protein's big. And you'll see what they look like today. It's a small molecule that can stop an infection once it's started. And, on every slide, I have a little quote in the beginning. And this one I really like. It's from Leo Tolstoy. Though the doctors treated him, let his blood, and gave him medications to drink, he nevertheless recovered. Okay, a few of you get it. I'm glad to see that. No biological is without side effects. Just remember that. Mostly it's okay, but none is 100% safe because people are all different. They react differently. There's nothing we can do about it. So last time we talked about vaccines. They can prevent disease, but they don't have much of an effect once an infection has started, especially an acute infection, right? Because they're pretty quick. There is one exception. Can you remember what that exception is? Where you could vaccinate even after you've been infected. Rabies, exactly. You have a couple of weeks before the virus gets into your brain and you can be immunized. So if you are ever bitten by a wild animal and you're not sure if the animal runs away and you can't give it a rabies test, you should get a rabies vaccine because you just don't know. In particular, if a raccoon is misbehaving. So let's say you're out on the front porch of your house, you're sitting there on a Saturday afternoon, and this raccoon comes up and bites you. That's not normal for a raccoon. Raccoons don't do that. It's rabid. Get a vaccine. Why am I telling you this? It's just interesting, right? So most of the time, if you're not vaccinated, or you can't be vaccinated, or you choose not to, and you get infected, you can take an antiviral, and that's what we're going to talk about today. However, we don't have a lot of antivirals. This is a time course of antiviral discovery over, the last 50 or 60 years or so. Most of them are against HIV, hepatitis C virus, herpes viruses. Why? Because these cause long-term infections. You have plenty of time to treat these. Whereas, SARS-CoV-2, you have a window of 7 to 10 days. If you don't get treated in that window, it's useless to use an antiviral. But these viruses reproduce in you a long time. Look, we have over 40 HIV drugs. Hep C, not so many influenza. That's one of the harder ones because it's an acute infection. But we don't have a lot. And on here, of course, aren't the SARS-CoV-2 drugs. We have a few of those that we're going to talk about today. Here's an interesting slide where we break down the drugs by virus and target. So you can see the pie chart on the left. Most of the antivirals are against HIV-1, then hepatitis C virus, then we have Hep B, and herpes virus is influenza. And then in the middle is the target, where what viral protein is the drug inhibiting, and that's the vast majority are polymerase and protease, because they're enzymes, right? You don't have a lot of them around in the cell because they're catalytic, so you can easily inhibit those. And we have integrase, NS5A, and others. And then on the last pie chart, interesting, whether it's virus or host target. So most of the time, you can see 75 of the antivirals are targeted to a host protein, a virus protein. But sometimes we're able to make antivirals against the host. And we're going to talk about some of those today. They're somewhat interesting because, the idea is that if you target a host protein, maybe it's harder to get resistance, right? Because the host is DNA and lower mutation rates. But in fact, you can get resistance to host antivirals as well. So why so few? There are a number of reasons. And one of them, a big one, is that a compound, even though it inhibits the host, the virus can also inhibit the host cell. So you would have side effects, and we can only deal with so many side effects. Anybody ever take Paxlovid? I have. Do you know what the side effect is? One side effect, you get this weird metallic taste in your mouth for the time you take you shouldn't take Paxlovid because you're too young and healthy, but my doctor, Daniel Griffin, told me I should. I wanted to see what would happen, but he didn't think it was a good idea. Maybe I would never have come back to class. Oh no, it was last November, yeah, if I had died, but it's got this very weird metallic taste. But we'll talk a little bit about that. So side effects are not good. That's obviously a side effects that you can deal with. And of course, viruses require host functions, and the antivirals can inhibit any of those accidentally. We call those an off-target effect. So that's very hard to get around. I don't want to minimize it. To find a drug that doesn't target the host is very difficult. But the second part is that many viruses are hard to work with. You can't grow them in cells in culture, which makes it really hard to find an antiviral, because that's usually the first thing you do is put compounds in cells and see if they have an effect on reproduction. And then there are problems with animal models. Smallpox, there's no animal model for studying smallpox, and yet the US a number of years ago wanted to make 2 antivirals and stockpile them in the case of an outbreak, because smallpox is a great bioterror weapon, technically, because it spreads very effectively. It's 30% lethal, and we're not vaccinating against it any longer. So someone could infect themselves with smallpox virus, walk into a major city like New York, and just stand in Grand Central Station for a day and infect tens of thousands of people. So the US had 2 antivirals made, and they had to get around the rule of having animal models because there weren't any. And then some of them are very dangerous, like Ebola and Lassa virus. You have to work with them in a BSL-4 lab, the highest level of containment, where you have to wear a spacesuit with an air hose attached to it. It's very difficult to work in those conditions. So this inhibits the production of antivirals. But the third reason why we have so few is the most important, and that is, to be useful, a compound has to block virus reproduction completely. It can't be 30 or 40 or 50%. You know, an aspirin is not 100% inhibitory of its target. It's something less than that, but that's good enough to get rid of your headache. And that's the same with a lot of standard pharmaceuticals. They're not 100% inhibitory, but antivirals have to be, because if you have a less potent inhibitor, you will select for resistance. And so here's a graph showing you using 3 different concentrations of an antiviral and monitoring viral load with time. And the optimal dose completely inhibits reproduction, and then an intermediate and low dose allows for reproduction. And reproduction in the presence of a drug, that's a great way to select for resistance. So you need to inhibit almost completely, and that's hard to do. The other issue, of course, is that at least for acute infections, they can be short in time, and often in these infections, by the time you have symptoms, by the time you feel ill, it's too late to give you an antiviral. Not the case with all infections, but influenza, for example, the antivirals we have, you have to take within a day or 2 of feeling those first symptoms of influenza. Otherwise, the drug won't help you. And it's different for SARS-CoV-2, as we will see. Now, you may say, well, why don't we give people drugs ahead of time and just prophylactically treat them, right? That's what prophylaxis means, to treat them ahead of time. And that's usually not a great idea, especially if there's some side effects to the drug. But there is one exception. We do pre-exposure prophylaxis for people who are at risk for HIV infection, and you can prevent infection. So that's a good use of that. But you don't need to take the drug all the time. You just need to take it when you think you're going to have an encounter. The other issue is we don't have any broad spectrum antivirals, so it's not like we can predict what you're going to be infected with, right? So if we had a broad spectrum, we could treat you with that, but we don't. Not yet, at least, although I'll tell you a little bit about that. And then the other issue is diagnostics. You need to be able to diagnose someone quickly and give them a drug. And that's hard, as we saw during the COVID pandemic. It took a while for us to get diagnostics up and running and make them rapid so it doesn't take days for you to get the results. You know, in the beginning of the pandemic, the CDC wanted to do all the testing. So if you were some clinical laboratory throughout the US, if someone came in with symptoms, you would take a sample, put it in a tube, FedEx it to them. They'd get it, do the test in a couple of days, and send you the results 4 or 5 days later. That's absurd, right? Totally absurd. I don't know why they wanted to do that, but they realized the folly early on and changed it. And then eventually we developed rapid assays where you could do it at home. You get a positive test. That's what I did. I had symptoms. I had one of these lateral flow assays. I did it. It was positive. And then you call your physician and say, hey, I have a positive test. And you get your drug. But that's not the case for most infections. All right, so those are some of the problems. Let's look a bit at the history of drug discovery. It really started in the 1950s. We knew that antibiotics were very good for inhibiting bacterial infections. Sulfonamide antibiotics were very successful. So chemists began to modify them and make compounds and test them against different viruses. Thiosemicarbazones were found to be active against pox viruses, and smallpox was still a threat after World War II. In fact, in 1947, I think it is, a man walked into New York City, I think Grand Central, that's why I mentioned that before, and he had smallpox. And he went to a hospital. He was diagnosed with smallpox because back then there was enough of it around that you would be aware of it. And I think New York City vaccinated millions of people within the next weeks to prevent spread. It never spread very extensively, but it was a big deal even back then. It was eradicated in 1979. Now, in the 60s and 70s, programs were begun at many companies, which we call blind screening. In other words, we just take mixtures of compounds and apply them to a virus-infected cell, see if they're inhibitory. So you could take dirt. You take some dirt, soil. Sorry, it's not dirt, soil is the right name. Soil, and then you can mix it with buffer and take an extract of it and put it on your cells. And you can find inhibitors. Then you'd have to purify whatever it is that's doing that. But that's called a blind screen, because you're not focusing on a particular virus or a virus-specific mechanism. You're just saying, does something in my mixture inhibit virus multiplication? And so you can see, you can collect dirt, soil, sorry. I just grew up with dirt. I'm sorry, I'm never going to get it right. Soil, and see if it has something in it. Usually there are various microbes in the soil that are producing antimicrobials that you're actually assaying. Many companies have huge collections of compounds that they've made for other purposes, and they keep a little bit of each one, and you can check all of those to see if they're inhibiting, and that's what people did at the onset of the COVID pandemic. So then you get a hit, something that inhibits your virus reproduction in vitro. Then you have to purify and get whatever it is that's active. And then the chemists step in and they modify it because you have to improve the properties of the chemical. You have to reduce toxicity. You have to make sure it's soluble. It has to be bioavailable. In other words, you take it orally. Does it go to the place where it needs to go to inhibit infection? In some cases, it's not bioavailable, like remdesivir, and has to be injected intravenously into you. And then there are other pharmacokinetic properties that we're going to talk about that have to be modified as well. And typically you make many, many thousands of molecules before you find something that's active. And an example is this drug called Symetrel or amantadine, which was approved in the 60s for treatment of influenza A viruses. And it's one of a couple of drugs we now have, although most of the influenza viruses out there circulating are resistant to this drug, and so we can't really use it. But its mechanism was not known when it was first discovered, yet it was still licensed. And we figured it out years later. This is the drug that inserts into the ion channel in the virus particle and blocks protons from going inside the particle. Remember, the protons will lower the pH in the particle and allow the ribonucleoprotein to come out. and get into the nucleus. So that's how this works. It blocks that channel. Today, antiviral discovery is very different. We don't do any of these blind screens anymore. We don't go out and collect dirt. You know, the divisions of these companies that used to do that were called natural products divisions, and they're mostly abandoned. But now we use different approaches. We can do very targeted drug discovery. We have recombinant DNA, so we can clone an essential gene like a polymerase or a protease. We can make the protein in yeast or insect cells. We can do structure analysis, and we can identify molecules that might inhibit it. Of course, since we know reproduction cycles of many viruses, we can decide what we're going to look at in the cycle for an inhibitor. And even if you can't grow a virus, you could find an inhibitor of the polymerase and get that, and then hopefully you have an animal model and you can test in preclinical studies if it works in them. So no more blind screening like I just described for you. Here's our standard reproduction cycle of a virus. And on this, I've put a number of viruses for which we have inhibitors at every step. Starting at attachment, we have attachment inhibitors, penetration uncoding, mRNA synthesis, processing using an inhibitor of a protease. And then, of course, we have many inhibitors of DNA and RNA replication. Polymerases are very popular targets. We even have an integrase inhibitor, multiple integrase inhibitors for HIV-1, assembly inhibitors, and even release. Every step of the cycle, we have some inhibitor. This is a testament to scientists studying the reproduction cycle and understanding what's going on, and then being able to select for drugs or identify drugs that inhibit them. So here's an overall look at the drug discovery pathway. You know, typically it takes 10 to 20 years to identify antivirals. We were very lucky during the COVID pandemic that some of them were identified in a year or two. So you start with a medical need. You have to have a lot of people infected. You do some research to figure out a target. So we say, okay, this virus encodes an RNA polymerase. We're going to go for that. And then you have to have a proof that that's going to work. In the case of an RNA polymerase, it's pretty obvious. But if you have a less obvious protein, you could always remove the protein coding region from the genome and show that it's essential for replication. That would be really good to do. So that's what I mean by proof of principle. And then you can do some structures and get some idea of what the protein looks like and maybe design inhibitors to go into that. You could screen libraries of inhibitors, natural products, which we used to do, then collections of compounds. You can make collections by combinatorial chemistry, RNAi. And there are companies today that sell inhibitors, and they can tailor them to exactly what you want. If you want an inhibitor, say, of a protease, they will have a collection of drugs that are designed to fit at the protease active sites. If you want an inhibitor of transcriptional activators, they will have a library of compounds just for that. It's really remarkable. Then in the middle, you do your screens. They can be cell or mechanism based. I'm going to explain that to you in a moment. Or they can be in silico. You can have a computer say, here's the active site of the enzyme. Let me tell you what fits into it. And a number of drugs have been designed that way. And of course, you have to make the compound and show that it works. You have initially compounds that are inhibitory. You have to modify them, of course, to make sure, for example, they get to the right place in the body at the right time. Bioavailability, pharmacokinetics, will it last long enough? If it goes away in a few hours, that's not useful. And will it be safe? You have to do toxicity and specificity, first in cells and then in animals. And all of this work in cells and in animals is called preclinical studies. And then assuming that looks good, you make an application to the FDA, and then you go into people. Clinical testing means people, of course. It takes a long time, and it takes a lot of money. And when we do clinical testing, we talk about phases of a clinical trial. So here's a timeline. Look at, we're going out to 20 years here. There's your discovery and synthesis, preclinical studies in animals. And then you have a phase 1. This is typically a safety analysis in 20 to 80 healthy subjects. You know, they can't have any illnesses. And you just want to know if the doses you're thinking about are safe. And you have a pretty good idea because you put this in animals at very high levels. You basically dose animals until they drop, and then you back off quite a bit. So you know the mgs per kg of what would kill an animal. And then the phase 1 data are collected. You write them up, you give them to the FDA, and then sometime later, can be a year later, they say, okay, you can do a phase 2. Then you do a phase 2, which is efficacy in a little more number of subjects. Same thing, you get your data, you give it to the FDA, they look at it, and then they give you a go-ahead for a phase 3, which is efficacy, side effects, and superiority to what you have. And these can be very big studies. This was all shortened during the COVID pandemic. They overlapped, phase 1, phase 2, phase 3. As soon as you are completing phase 1, looks good, we're going to go right into a phase 2. Otherwise, many, many people would die. FDA review, and then can be given an emergency use authorization if there's an emergency like a pandemic, or it can be licensed, which means it can be prescribed outside of an emergency. And once your drug is licensed, you continue to collect data. That's ongoing safety surveillance for many, many years. All right, so that's why drugs are expensive and it takes a long time, but in an emergency, we can get around that. So let's look at some screens. People can be very clever to devise screens for looking for antivirals. This is a mechanism-based screen. In other words, here we're looking for an inhibitor of a protease. That's the mechanism. It cleaves a protein. So we identify the cleavage site on the protein for the protease. That you need to do some science to do that. And then you make a substrate. This is just a peptide with, at one end, a molecule that will make some kind of signal. It will glow or something that you can measure. And then there's a bead attached to the other end. And so when the protease is mixed with this reagent, it will cleave the two. It will separate the fluorescence from the insoluble part. The bead can be removed by centrifugation. And if you have an inhibitor, you will block that. And so here's an example. We're having time in minutes on the x-axis and fluorescence intensity of the soluble peptide. Remember, the peptide is soluble with the fluorescence marker on it because the bead is on the other half. So you can do the reaction, centrifuge out the beads, and then measure their fluorescence. And you can see the reaction is very quick without the drug. And then you add the drug, you have an inhibition. So you can do this with lots and lots of compounds. This is a kind of assay that lends itself to what we call high throughput formats, doing tens or hundreds of thousands of compounds in a day. Here's a cell-based screen where we're looking for an inhibitor. In this case, it is a protease inhibitor, but we're using cells instead of solution. So here we have bacteria, E. coli typically, that are engineered to be tetracycline resistant. So they produce this membrane crossing protein that gives them tetracycline resistance because it pumps the drug out of the cell. And this has been engineered to include an HIV protease site on one of these loops inside of the E. coli cell. And so if you treat these bacteria with tetracycline, they will grow in the presence of tetracycline. If you produce the HIV protease in the cell, along making sure you have the cleavage site in this protein, this will not be resistant to tetracycline. They will be inhibited. And so no colonies will arise in the presence of the protease. But if you have an inhibitor of the protease, with this red blob blocking the Pac-Man there, it will not cleave the pump, and these bacteria will be tetracycline resistant. So you can use colony numbers as a readout of the effectiveness of your inhibitor of the protease. So these are also very clever and high throughput approaches. We can do many, many compounds a day. We can look at chemical libraries, as I said, natural products still to some extent. Combinatorial chemistry. Chemists take various linker molecules of sorts and fragments of compounds, and they can mix them in matrices like this and make thousands of random designs, and these can all be tested for activity. You can have the structure of your protease, say the active site, and have a computer tell you what kinds of molecules will fit into it. You can make those and then test them in whatever your assay is. And all this is done by robots now. We have micro titer plates with many wells, 96, 384, 1,536 wells, and in each one you could assemble your reaction and put a different inhibitor in each one, and you can have the robots do it. These are robot arms in a screening facility. They will pick up the plates individually, and they will also put compounds in the wells, as you can see here. They will put them into an incubator, and then the incubator will read out the reaction in every plate. So the whole screen can be assembled robotically and done robotically. And then, of course, the results are uploaded to the cloud, and you can look at them wherever you are. Sitting on your front porch with a rabid raccoon, maybe, you could look at the results. So we can do lots and lots of screening of many, many compounds to find the rare ones that are going to inhibit. Question number 1, we have many antibiotics but fewer antivirals. What's the reason for the difference? A, robotic screening is slow. B, there are few serious viral infections. C, resistance is a problem. D, antivirals must be potent. All of the above. Why do we have so few antivirals? How do we do? Antivirals must be potent. That's right. Some of you picked resistance. So it doesn't limit the development of antivirals, but it makes them not useful at some point, right? So we have a lot of antivirals that are still out there for which we have a lot of resistance. So that's not why we have fewer antivirals, just because they have to be potent of all of these. The screening is not slow, and there are few serious infections, of course, isn't correct. Unfortunately, none of you picked either one of those. So that slide did mention resistance. So let's talk about resistance and how it works. And if you make a drug, you should assume that you're going to get resistance to it, because viruses reproduce very efficiently, right, very quickly, and they make a lot of progeny, and they have pretty good mutation frequencies from DNA to RNA viruses. And this resistance is a problem, especially if you're treating patients for a long time with antivirals. So you would have a chronic infection like HIV or Hep B or Hep C even, although we've reduced the treatment time for Hep C now, so it's not as much of an issue. But we have resistance to everything. All of our drugs that we use, we have some extent of resistance to them. And this is a problem because we don't have a lot of drugs. So if you're treating a patient and patient then suddenly has a virus in it that is resistant, then you may be in trouble, because if you don't have a lot of other antivirals, you can't treat them any further. So you can't treat the patient with the same drug if resistance arises. So HIV/AIDS patients are periodically monitored. You get an isolate from them, and you do sequencing to see if there are any resistance mutations. In fact, you do that before you even treat a patient, an HIV/AIDS patient, with drugs, because you want to give them the right drugs. If they're having already resistance to the drugs, there's no point in giving it to them. And if you don't have any alternative, you can't stop the infection, which is not a good thing. However, we can look at resistance and understand how it arises, and sometimes we can design new drugs that have a less frequent ability to select for resistance. And we've done that for a number of drugs. In fact, early in the pandemic, a number of laboratories were selecting SARS-CoV-2 resistant to remdesivir. And a lot of people said, why are you doing that? These viruses are going to get out and become a problem. Well, in fact, you do it to see if you can get resistance, and then maybe you can design the drug to get around that. So it is a worthwhile thing to do. And so the mechanism of resistance is that polymerases are error-prone. RNA polymerases, all polymerases are error-prone, but RNA polymerases have no correction mechanism, with the exception of one order of viruses, which I'll tell you about. They make a mistake in every 10,000 to 100,000 nucleotides polymerase, which is about 1 million-fold greater than that of the host genome, very high mutation rates. And so in a 10,000 base RNA genome, you get a mutation in every 1 to 10 genomes. And that's a lot, because you can make 10,000 viruses in a cell, which means you have the whole range of mutations present. So you make random changes, and then one of them may give rise to resistance just randomly, and then if the drug is present, it will select for that. Now, the nidoviral genomes encode a proofreading exonuclease. This is unique among RNA viruses. No other virus encodes this. So here's a picture on the bottom of the RNA synthesis complex. These, of course, include the coronaviruses. So nidovirales is an order, and an order is composed of different families, and the one here is the Coronaviridae family, in which we have our human and animal viruses. All of these encode a exonuclease. It's called NSP14. And here's the RNA-dependent RNA polymerase in red, and then a lot of other proteins that are needed for RNA synthesis. But essentially, the exonuclease, if there's an error, the exonuclease cuts out the wrong base, and the polymerase will repair it. So this reduces the error rate about tenfold. It still has a high error rate, but it's lower than most other RNA viruses. And the reason we think these viruses have to encode this is that their genomes are so long. They're 30,000 bases and bigger. And so without a exonuclease, those big genomes can't maintain viability. In fact, you can do the experiment. You can remove the exonuclease from the genome. The virus will still reproduce, but it has really bad fitness, and it can't compete. It would never be able to transmit in population. So this gives it the ability to maintain a relatively error-free genome, not error-free, but lower error rates, and it can be that long. And DNA viruses, though, they do have an error correction mechanism. They do have exonucleases, and so they can be corrected. So here's an example of a DNA polymerase making a copy of a single strand of DNA. And there's a mismatch happening here on the left. You can see that the two bases are not pairing. And that happens because the polymerases are not perfect. The exonuclease will detect it, shoo it out, and then remove it, and then the polymerase will fill it in and correct it. So this reduces the mutation frequency of DNA virus substantially. But as a consequence, DNA viruses evolve much more slowly than RNA viruses. And we'll talk about the consequences of that in a bit when we talk about evolution. But anyway, these random mutations give rise to changes in the genome. And if you apply a drug, and there happens to be a change in that population of viruses that makes resistance, that virus will have a fitness advantage and will outgrow all the others. So I want to go through now all the steps in virus reproduction and give you examples of inhibitors at every step and how they work. So first we'll start with entry. Amantadine, which I mentioned, one of the first antivirals to be discovered. That's the structure of amantadine, a very odd three rings joined together with an amine. And this interacts with the viral M2 protein, which is an ion channel. Remember, during the entry of influenza virus, there's the endosomes become acidified by the entry of protons into the endosome. Those protons then flow into the interior of the particle through this ion channel, allowing the RNAs to get out. And so here's a depiction of the ion channel. It's made up of 4 molecules of the M2 protein, the same one that's the target for amantadine, and the ions will flow through into the interior. Amantadine is shown here. The structures interacts with specific amino acids. Here's the red amantadine there and on the outside as well. And it prevents the protons from going through into the interior. So it effectively blocks proton transport. So it blocks infectivity. The viral RNPs cannot get out of the particle. But we have extensive resistance to that already, so it's not really used. Another entry inhibitor is Maraviroc. It's an inhibitor of CCR5. Remember, HIV-1 needs 2 receptors to enter cells. It needs CD4 and CCR5. So the way it works, the glycoprotein of HIV first binds CD4. That causes a conformational change in the GP120 subunit. GP120 then binds CCR5, and that causes the fusion peptide to flip out. But Maraviroc is a small molecule. There's Maraviroc's structure right there on the left in panel A. And on the right is the structure of Maraviroc bound to CCR5. So CCR5 is in red, Maraviroc is in blue there. And it binds CCR5 and prevents it from binding GP120. So the presence of that small molecule at CCR5 blocks entry by binding and preventing fusion, essentially. I think I've shown you this, but it doesn't hurt to show you this again. This is an interesting story of why hydroxychloroquine could never have worked. Hydroxychloroquine was known for years to inhibit infection by many viruses by inhibiting endosome acidification. So hydroxychloroquine will go into the endosome and keep it neutral. And so many viruses who require acidification, their reproduction is inhibited. So a group of scientists, initially in Australia and then other places, did an experiment using cells in culture and found that SARS-CoV-2 reproduction was inhibited by adding hydroxychloroquine. And so the FDA was bullied into approving this drug, into giving it an EUA. It was bullied by the administration. Remember who was in charge back then, right? So they made the FDA approve it with no clinical trials in people. This should never happen, okay? This should never happen. You need to test it in people, because when it was finally tested, it did no good and it actually harmed people. It didn't work. It was given an EUA by the FDA, which was subsequently retracted. Why didn't it work? Well, the cells that they used were kidney cells, and kidney cells are susceptible to infection, but they do not allow entry in the same way as do respiratory cells. In kidney cells, remember, in the lung, the virus binds to ACE2, and then there's a protease on the plasma membrane, Temperase2, which cleaves the spike, and then the virus enters at the plasma membrane. This pathway is not inhibited by hydroxychloroquine. Hydroxychloroquine has no effect on that. So the virus gets into cells quite well in the lungs in the presence of hydroxychloroquine. It worked in kidney cells because there's no surface Temperase2 there. The virus goes into the endocytic pathway, and there the hydroxychloroquine inhibits the activity of cathepsinella protease that cleaves spike there so that it can fuse. But, you know, they used the wrong cells, and they didn't think of basic virology, and neither did the FDA apparently in the administration. So this is really a sad story because a lot of people were treated and died because this had no effect on their infection. It's also a harmful drug as well. All right, so then we move on to polymerase inhibitors. We have a lot of those, and these are the 4 bases there in the yellow: adenosine, guanosine, thymidine, and cytidine. And for T, if this were RNA, it could be U, of course. And you can modify all of them in ways that inhibit nucleic acid synthesis. So let's take a look at acyclovir, which is a really good anti-herpes drug. Here's acyclovir in the blue box, and it's a derivative of guanosine. You can see that the ribose has been a bit wrecked, and there's another derivative of it called ganciclovir, which is also an inhibitor. So these two are drugs that are used to treat herpes infection. But you can see derivatives of all of the bases are made, and all of these drugs on the outside here are various inhibitors for various viruses. We'll talk about AZT, for example, in a bit. But many of these antivirals are either nucleosides, which means there's no phosphate. You see here, there's no phosphates on them, or nucleotide analogs. So acyclovir is a nucleoside analog. It inhibits herpes viruses. So how it works is as shown here. You give the drug to someone, it gets into their cells. Without a phosphate, things can get into cells very readily. If you put a phosphate on it, then it's a real problem. So acyclovir is taken up, and if your cell is infected with herpes simplex virus, one of the viral proteins is called TK, thymidine kinase. It adds a phosphate to acyclovir. If your cell is not infected, it is not going to phosphorylate acyclovir, and it will have no effect on the cell because this only works when it's triphosphorylated. The cell then takes over and puts 2 more phosphates on because you need to have 3 phosphates on the nucleoside to be incorporated into DNA. Then this base is incorporated by the viral DNA polymerase into cellular DNA. It's also incorporated by the cellular DNA polymerase, right? But the viral DNA polymerase puts it in DNA, and it's a chain terminator. because the next base cannot be added to it. So you see here, guanosine, the next base would be added to the hydroxyl there, but there's no hydroxyl on acyclovir. And so it gets incorporated, and that's the end of the synthesis, so it inhibits DNA replication. This is an interesting example of how to improve an already good drug. Acyclovir wasn't particularly bioavailable. In other words, when you took it orally, it didn't effectively get to the right place. But chemists figured out by just adding a valine to acyclovir to make valacyclovir, this is really bioavailable. You take it by mouth and it goes to where it needs to go, and then it gets into the cell. The valine is cleaved off by cell enzymes, right? And then it becomes acyclovir, and then it can work as it did before. So an interesting example how a very simple modification can improve bioavailability. There are, of course, acyclovir-resistant herpes simplex viruses that arise, especially in people treated for long periods, but this is still a well-used drug. And it's now over the counter, and you can buy it in little tubes. If you get a cold sore, you could just put it on it, and it works pretty well. There are two kinds of mutants that are resistant to acyclovir. Some have changes in the viral thymidine kinase genes. They can't phosphorylate acyclovir, right? So the polymerase is resistant because the acyclovir isn't phosphorylated, it's never incorporated. And then there are changes in the viral DNA polymerase gene that make it unable to recognize acyclovir itself, and so there's no inhibition. So those are the two kinds of mutants that arise. Another kind of nucleoside analog is azidodioxythymidine, or AZT, the first HIV drug. This was originally known in screens for anti-tumor compounds. And so, when HIV/AIDS first arose, they were trying everything to find inhibitors because they had nothing. So they tried AZT, and this worked, so this was used. So here's AZT, which is a derivative of thymidine, and it has a 3-nitrogen here, as you can see, instead of a hydroxyl. This is taken up into cells. It's phosphorylated by cellular kinases. You can see 3 different kinases here put on 1, 2, and 3 phosphates here. And then this is incorporated into the DNA by reverse transcriptase. It's a chain terminator. Again, you can't add the next base because there's no hydroxyl there, and that stops synthesis. And this isn't recognized particularly well by cell enzymes, so it's more selective for RT, but it does have side effects for sure. And this is the subject of this movie. If you've seen this, it's all about AZT. Yes, this is a story of how there wasn't enough drug to go around, so they split the pills, and it worked. And then NIH said, oh, we don't have to give as much, and we can give less. So the guys who were doing this actually helped them find the right dose. It's a really good movie. Anyway, it has a lot of side effects. It's given orally, but the half-life is an hour. 1 hour. So you have to dose 2 to 3 times a day. You feel like crap, so you stop taking it, and it's not going to work, of course. And if you do take it, because of the short half-life and multiple dosing, you're going to select for resistance. So within a year, there was resistance out there, and that wasn't very useful. Interesting story. So the resistance comprised single amino acid changes at one of 4 sites in RT. Here's reverse transcriptase. The active site is yellow. There is a molecule of RNA and DNA coming out of the enzyme. So single amino acid changes prevent the drug from working. The RT doesn't bind the phosphorylated AZT. And so very quickly, this drug wasn't useful, but new nucleoside analogs were developed right after that, DDI, DDC, D4T, 3TC, and you would get resistance to these right away. So they decided to start combining them, put 2 together, but still less than a year, you would have resistance to 2 of these together. Then the next class of inhibitors are called non-nucleoside inhibitors were developed. So these are not targeting the active site, they are targeting somewhere distant. So these red balls here are a binding site for nevirapine, which is this inhibitor. It's away from the active site, but it causes a conformational change so that the reverse transcriptase doesn't work. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And of course, we got resistance to these when used alone. And so we have a number of these, as you can see here. And So basically, none of these can be used effectively alone, and we realized that later, and we now use combinations of 3. And that's been the key to taking care of this infection for your lifetime, essentially. And we'll talk about combination therapy in a bit, but I want to talk about some of the SARS-CoV-2 drugs. There's a nucleoside analog, remdesivir, which is a prodrug of adenosine. So here's adenosine here on the bottom, and here on the top is remdesivir. So it's adenosine with this big group hanging off of it. And you see there's a phosphate there. So the chemists figured out how to hide the phosphate with other groups so that it would be taken up into cells. This is really interesting because otherwise you can't have a phosphate on. And that first phosphorylation step in the cell is very inefficient. So if you can already have a phosphate on there, it makes your drug more effective. This drug was developed in 2013 for the Ebola virus outbreak, but it didn't work very well. And then it was found to inhibit SARS-CoV-2 at the onset of the pandemic. It is a chain terminator. It's incorporated into the RNA. This group here is taken off in the cell, so you just have the phosphate. It was given an emergency use authorization after phase 3 trials, as it should be, but unfortunately it has to be given intravenously. And so it was largely, in the beginning, given to people in the hospital. And it doesn't work once you're hospitalized. It's too late. If you're in the hospital, it means your viral phase is almost over. You're in the inflammatory phase. You can't breathe. You need oxygen. And antiviral is of no use. Only when they started to give it to people early, before they were hospitalized, did it cause an 87% reduction in progression to more severe disease. And so the only negative about this, well, first of all, it has to be given within 7 days of symptom onset. That's not bad. It's intravenously delivered. Really hard to get it to a lot of people. All right, and then we have another nucleoside analog, molnupiravir. This is a prodrug of cytidine. And here, these are compounds on the way to molnupiravir. Here's EIDD 2801 is molnupiravir. And it basically is a mutagen when it's templated as a U. And so it causes a base change, and it was found early on to inhibit SARS-CoV-2 replication. And it is orally bioavailable, so it was given in EUA after a clinical trial. However, it has really modest effects now. I mean, the best it ever did was 30% reduction in progression, and now it's probably even less. And so it's a last resort. If you can't take remdesivir or Paxlovid, this is what's left, but it's not very effective. All right, next question is, resistance to which antiviral would involve amino acid changes in a viral enzyme? Acyclovir, amantadine, penicillin, or all of the above? All right, so we're looking for resistance conferred by amino acid changes in a viral enzyme. Okay, let's have a look. Acyclovir targets polymerase or TK in terms of resistance, so they're both enzymes. Amantadine, the channel is not an enzyme, and penicillin is, of course, not an antiviral. So it's just that one. Amantadine is not an enzyme, right? So that's the key to this question. All right, moving down the pathway, we've just done nucleic acid inhibitors. Now we have we have multiple integrase inhibitors. Remember that the retroviral DNA, once it's made by reverse transcription, is going to integrate into host DNA. It's carried out by a viral enzyme integrase. And there are a number of inhibitors, raltegravir and dolutegravir. And here is how it works, basically. This is a close-up of the active site of the integrase. Here's the 3' end of the DNA, one of the 3' ends. And this is, the integrase will take that and insert it into host DNA. So there's no drug. Notice the A residue here, right near the 3' end. And here now is raltegravir in magenta, sitting in the active site. Look what it's done to the A. It's completely pushed it down. And the same thing with dolutegravir in cyan. It's also pushed it down. So it inhibits the activity of the enzyme by doing that. Hepatitis C virus, we have a number of inhibitors for this. Here is one called sofosbuvir. This is a prodrug. And again, it has got the phosphate hidden there by a bunch of other side chains. So here is the nucleoside. Now, normally, again, the phosphorylation of the nucleoside to the monophosphate is very slow. And so if you can already put the phosphate in the drug, it speeds it up. But monophosphorylated compounds don't get effectively into cells. But as I said, the chemists figured out how to shield them. So now you can put this in. It's very rapidly diphosphorylated. And now this is the active triphosphate, which gets into the growing chain and inhibits it. It's a chain terminator. This is very expensive. One pill is $1,000. You know, you get your bottle and you take a pill and one falls on the floor all the time, right? It's $1,000 you have on your floor that your dog may come and eat up. You better watch it. 12-week treatment, which is what you need, $84,000. Wow, that's a lot of money. We'll actually get back to that. Baloxavir is a new influenza virus antiviral. It is an inhibitor of the endonuclease. Here's baloxavir on the lower left. My colleague Dixon Despommier calls this dihydroxy chicken wire. Doesn't it look like chicken wire? Yeah. Although there's no dihydroxy here. Difluoro chicken wire. Anyway, remember the synthesis of influenza mRNAs is primed by a piece of mRNA which is taken from host cell mRNAs, and the viral endonuclease cleaves the mRNA to produce those primers. So this drug was discovered as an inhibitor of the endonuclease. So it's another one in addition to imantidine that we have. We'll look at some more in a moment. Now, protease inhibitors are a big class of enzyme. Here is assembly of a retrovirus, and you may remember that the GAG-Pol precursor has embedded in it a protease PR, which gets incorporated into the particle. And then once the particle buds off, the protease cleaves all of these individual proteins from the polyprotein to make the mature viral particle. So it's absolutely required for the production of infectious virus particles. And so a number of inhibitors have been designed. And here is an example of a protease inhibitor that was designed based on the substrate. So here's the substrate for cleavage here. And then they modeled transition states, and they modeled the drug based on this transition state. It wasn't so active. They modified it further, and they ended up with ritonavir, shown here, which is basically a mimic of the polypeptide. So this looks like the actual substrate, you can see here, and it binds into the protease. So on the right here is a model of the protease with the active site in yellow, and the ritonavir is in red. So the ritonavir binds in, but it binds very tightly, and so it inhibits the enzyme. Now, if this sounds familiar, ritonavir turned out, besides being a protease inhibitor, it was an inhibitor of CYP3A, a mitochondrial enzyme that's involved in drug turnover. And it turns out that ritonavir can extend the half-life of many drugs, which are normally degraded by CYP3A. So ritonavir is given together with nirmatrelvir, which is Paxlovid. Paxlovid is 2 drugs. Nirmatrelvir has a short half-life, but if you give it with ritonavir, it extends the half-life and makes it very effective. So this is pretty cool that this originally developed as a protease inhibitor for HIV is useful for many other applications. Now, if you're taking drugs that you can't have too much of, you cannot take Paxlovid. Let's say you're taking statins, where the level of the statins cannot go up. You can't take ritonavir because they will increase the half-life and have you have poor side effects. We also have a hep C protease inhibitor. The hepatitis C RNA genome is translated to a long polyprotein that's cleaved by these proteases, including NS3. And the inhibitor of NS3 is telaprevir. And it's, again, a peptidomimetic. It fits into the active site of the protease. It looks like the substrate, but it's different enough so that it's locked in there and inhibits the protease. And of course, we have Paxlovid, which is an inhibitor of the SARS-CoV-2 protease Mpro. The genome is translated into a long polyprotein here on the left end, and it's chopped up by a protease. So an inhibitor was identified, and its chemical structure is shown here. Again, a peptidomimetic. And on the right are two activity profiles of this protease inhibitor. So on the top is a measurement of the enzyme's velocity, of its ability to cleave substrate. And what they're looking at here are proteases from different coronaviruses. So we have SARS-CoV-2, SARS-CoV-1, a couple of common cold coronaviruses, and MERS coronavirus. And it's best against SARS-CoV-2. That's the black line there. So the 50% velocity is at about 10 nanomolar. And it does inhibit some of the others, but you need more concentration. So this could be used in an emergency if a new coronavirus pandemic emerged at some point in the future. This could be useful. But if you look at inhibition of viral CPE, you can see that all these viruses are similarly inhibited. So that bodes well for treatment of people. We have release inhibitors for influenza virus. Now, at the end of the influenza reproduction cycle, remember the viruses bud from the surface of the cell and they're released. Well, if the cell has sialic acid on it, the viruses are just going to stick to it. Because remember, sialic acid is the receptor for the virus. So all these viruses would stick to the cell unless there's a neuraminidase present, which is in the virus particle, which will cleave off these sialic acids and allow the viruses to go free. Structural chemists took the structure of neuraminidase, which is shown in B, complexed with sialic acid, shown in green there. So that's the normal ligand of the neuraminidase. And they said, let's make molecules that look like sialic acid. They had the computer do this by in silico design, and they came up with these two compounds, Relenza and Tamiflu. And you can get either one of these. Tamiflu, you take a pill. Relenza, you inhale. But they're both inhibitors of neuraminidase. And if you treat infected cells with these compounds, the viruses stick to the surface. After they bud out, they stick because the cells and the viruses are covered with sialic acid, so they're sticking to one another. So that's how they inhibit infectivity. They are inhibitors of release. You do get resistance, but these were designed to mimic sialic acid. And the idea being, if we're as close as possible to sialic acid as possible, if the enzyme changes to be resistant to the drug, it's also not going to bind sialic acid, so those viruses are not going to be infectious. And that has almost been verified. So here is a cartoon of neuraminidase in purple binding sialic acid. It's going to cleave it off the cell so the viruses can be released. Oseltamivir fits into the active site, but zanamivir looks more like sialic acid than does oseltamivir. It's easier to get resistance to Tamiflu than it is to zanamivir because Tamiflu is a little bit different from sialic acid. Very hard to get resistance to zanamivir. So it's an interesting lesson in you design an inhibitor to be as close as possible to the ligand, and that may make it harder to get resistance. All right, our final question is, which of the following targets for HIV antivirals inhibits the earliest step of infection? Nucleoside inhibitors, non-nucleotide reverse transcriptase inhibitors, CCR5 inhibitors, integrase inhibitors, or fusion inhibitors. The earliest stage of infection. Okay, what do we have here? CCR5, right. CCR5 is the earliest step. So nucleoside is not early, that's nucleic acid synthesis. NNRTIs is nucleic acid. CCR5 is binding. Integrase is further down. Fusion is right after binding. So that's close, but it's not the earliest. How about broad spectrum antivirals? Can we make one drug to inhibit them all? Maybe. So this is favipiravir, which inhibits a lot of RNA viruses, and its target is the RNA polymerase. There's favipiravir. It's taken up into cells, and it's converted to a base, a nucleoside or nucleotide that has one phosphate on it, and then it's triphosphorylated, and that's incorporated into growing chains, and it inhibits chain RNA synthesis. And you can see a lot of plus and minus strand RNAs viruses are inhibited by these. But the only licensure is in Japan so far to treat influenza. And I think it's just not potent enough to be able to be licensed in many other countries. We also have a broad spectrum inhibitor of DNA viruses, cidofovir. That's what it looks like. You can see the ribose is all messed up. It does have a phosphate on it, allows it to get into cells. And this is, again, the phosphate group makes it a mimic of monophosphate. It's then diphosphorylated and inhibits quite a few DNA viruses as well. So I want to end up by telling you two stories of antiviral success. This is combination therapy for AIDS and hepatitis C virus. So on the left is the reproduction cycle of HIV-1. We have inhibitors at multiple steps: attachment, entry, fusion, RT, and integrase, protease inhibitors. So we can combine some of these. And for hep C, we also know the reproduction cycle, and we have inhibitors available at multiple steps. And the key to drug development for these viruses is that they cause, in many cases, well, in HIV for everyone, lifelong persistent infections, and in many people infected with hep C as well. So with combination therapy, we call this highly active antiretrovirus therapy. We make a chronic disease out of HIV. It is no longer lethal. We usually have 3 inhibitors in a pill and targeting different mechanisms. It allows you to live with infection forever, but you're not cured. You still have latent virus, and you have provirus in your long-lived memory T cells, which you can't get rid of. And we'll talk about that in some detail when we talk about HIV/AIDS. So here are the numbers for drug resistance. If you need 1 mutation in the genome for drug resistance, and you have a mutation rate of 1 in 10,000, that means every 10,000 viruses made, 1 base is substituted. An HIV-infected person makes 10 to the 10th viruses a day. So that means 1 million viruses are produced every day with resistance to 1 drug. That's why you get drug resistance so quickly when you just use 1 drug. Resistance to 2 drugs is the product. So you need 10 to the 8th. That's, if you're making 10 to the 10th a day, 100 viruses are resistant to 2 drugs a day. So we still see resistance. But for 3, you need 10 to the 12th viruses, and that's really hard to achieve. And remember, these drugs do suppress replication, so you're not getting these ideal numbers every day. We do see resistance to triple drug therapy, and that's why we still have to check patients before treating them, but it's rarer than for 2 and 3. These are all the HIV-approved drugs that we have so far. These are all the different stages: RT, non-nucleoside protease, integrase, entry, and these are the years that they were developed. And then at the bottom are the combinations. We have combinations of 2 and mostly 3 of different inhibitors sold by different companies. And new combinations continue to be developed. You have to test them, of course, but they're always developed, and they tend to be effective. And when they're no longer effective, we can move to a new one. And this saves people's lives. This is with years, the actual and projected numbers of people receiving antiretroviral therapy by region. So Africa has the most HIV/AIDS of any continent, and so we were treating very few people initially, but now we are treating more and more, which is great because they need it the most, and other regions of the world as well. And this is the number of people whose lives have been saved by antiretroviral therapy. Here, 10 million total adult deaths averted by 2016, and mother-to-child transmission of HIV have been averted by treating the mother before birth. So these are all the new HIV infections in children acquired at childbirth, and those have gone down with treatment of the mother and the red line is the number of infections averted. All you have to do is treat the mother before birth. That lowers the viral load and reduces the chance of transmitting it to the kid. And now we also do pre-exposure prophylaxis. So post-exposure is when you test positive for HIV/AIDS and then you're treated, but it's too late. By the time you have symptoms, it's too late to cure you. But if you do pre-exposure prophylaxis, for those at high risk, it will reduce sexual transmission and it will reduce transmission by intravenous drug use as well. If you're IVD drug user, you can take this. Doesn't bring it down 100%, but it's better than nothing. And this is typically two drugs. You can see here Truvada is 2 different drugs combined. Hep C has also been taken care of with drug therapy, triple and double drug therapy. In the old days, we used to treat Hep C patients with interferon and ribavirin. So ribavirin is an inhibitor of the polymerase interferon. You know what that does. And these patients had to be treated for up to 80 weeks, over a year of treatment, and many of these patients didn't respond. And as you know, interferon makes you feel really sick. So a lot of patients didn't continue their treatment. And so new inhibitors were developed, boceprevir, telaprevir, which had to be used initially in combination with interferon and ribavirin. But then it was shown that they were very effective, so they are subsequently used on their own. And now we have double and triple combinations where we've reduced the treatment to 20 weeks for Hep C. Now you have a complete cure of Hep C because this virus doesn't integrate into the genome. It's an acute virus infection that can become persistent, but there's no integration. So you treat, you can get rid of the infection, and the patient is theoretically cured. The problem is that many of the Hep C patients are intravenous drug users. They continue their habit and they get infected again. So we still would like to have a vaccine for Hep C to take care of that. But these 2 drug combinations for HIV and Hep C have really made a big difference. And the problem with HIV, of course, not just that there is a latent reservoir in you that we can't cure with drugs, but that there are so many genomes on the planet. We have 10 to the 16th HIV genomes. That has already resistance to everything we have and everything we'll ever make in terms of antivirals. So it would be nice to have a vaccine, but that just hasn't worked after many tries. All right, next time we're going to talk about evolution of viruses. Where do new viruses come from and where are they heading?